Okay, so now, guys, the next thing that we're going to see is, see, this is what we were talking about, like, how we are going to get, like, better yield. So first we were talking about, like, what really happened in your green revolution, what were the rice varieties, what were the wheat varieties, the semi-dwarf. Okay, these are the semi-dwarf ones. So how did he work with this thing and how he increased the productions? Then we start talking about your sugar cane. Then finally, just to end off with that, we talked about your millets. Okay, so that's it. That is what we talked about with regards to your plants. But now, the next thing is, guys, we are going to talk about, like, you know, we are going to develop plants against disease resistance, meaning against certain kind of your diseases. So let's try to see how is that really going to work. What do we really need to know? Just a second. Yes. Okay. Kindly ask your doubt. No issues. Go ahead. Millets, basically, millets are nothing but, especially when we talk about your small, like, this thing, like a ragi, jawar, bajra, like small grains. Like grains also, we have, like, very, very small grains. So those are nothing but your millets, actually. Okay, especially if you have rawa, ragi, and all, that is also, yes, it is also considered in that also. Okay, but generally when we use the term millets, it is that. Okay. Okay, sure. But now the first thing that we need to know, guys, is, okay, we are going to develop plants against certain diseases. Okay, so plant breeding for disease resistance. Okay, but now, if, before we talk about, like, what is what are the different types of like, plants that are going to be breeded for, like, disease resistance. First of all, we need to know what is the threat from different kind of your bacteria, viruses, or even your fungi. How are they going to affect your plants? So guys, let's try to see this thing. And guys, this is very, very, very important from your need point of view, guys, because they will ask you, okay, which is the fungal disease, which is a bacterial disease, which is your viral disease. So guys, once again, so what are we going to have in your fungal? We have labor. Always remember labor. Late blight of potato. We have brown rust of wheat, and we have red rot of sugarcane. Actually, guys, in this red rot of sugarcane, when the sugarcane is affected by this fungus, when you cut down the sugarcane, what do you see inside? Inside, you are going to see that, it is reddish like blood. That is what is going to happen. Sometimes you must have seen this brown rust of wheat. Okay, if sometimes, you store wheat, and, if it is like, infected, then what happens? You see some, a powdery substance coming out of it. That is nothing but your this thing. Okay, that is nothing but your brown rust. And in the case of your late blight, in late blight, what happens is you're going to see that the flowers, they are not going to bloom like properly. It is going to be really, really delayed growth and delayed blooming. That is late blight. So these are your fungal diseases. Okay, but now the next thing that we have is your bacteria. In your bacteria, we are going to have black rot of cruciferous. Okay, black rot of cruciferous. So guys, my question to you is, you must have bought cauliflower at your places, right? So cauliflower, let's imagine this is your cauliflower. So this is the lower part is green, and you are going to have this as the white part. Okay, now sometimes these cauliflowers, they are like really, really white, ekdam safed ki chamkar. Okay, so that way. But guys, have you ever noticed something? On top of these, sometimes you have these black dots. Okay, these black dots. What do we think? Arey, 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 ये तो धूल लगा रहेगा. Something must have happened this way. No, guys, this is actually black rot of cruciferous. Okay, what we do generally, we are going to just remove that portion off. Okay, so whenever you are making bhaji out of this thing, 1 minute, someone is asking what. Okay. Okay, so we don't use this part. We generally cut this thing off, and then only you are going to use that cauliflower. But then this is nothing but black rot of crucifers. Okay, and then finally we have your viruses. In viruses, we have tobacco mosaic virus. This is the most famous one, guys. Okay, mosaic virus. What does it do? It causes yellowing of tobacco leaves. Okay, and then the tobacco leaves are going to be like really really bad. So that is tobacco mosaic virus. Then we have something on the same lines, that is turnip mosaic virus. Okay, so both of them, they are on the same lines. Okay, causes yellowing of the leaves, and infect the leaves especially. So that way. Okay, but now guys, the next thing. Okay, the next thing we are going to see is, so we have these diseases. Okay, generally these diseases, but we are going to develop plants to overcome these diseases. So what are the plants and what are the different names? So let's try to see what are these disease resistance plant. Okay. Okay, so now guys, we have different types of your disease resistant plant, and this is what you need to know. Okay, in your exams, guys. Okay, once again, from your need point of view, once again, very very important. Okay, first of all, we have wheat. Wheat, it is himgiri. Okay, the name of the variety is himgiri. And what is this going to be like a resistant against? Leaf and stripe rust and hill bunt. Okay, very very important, guys. Guys, just remember, if you forget, just remember himgiri, hill bunt. Okay, edge. Just remember it this way. Then we have your this thing, the brassica. Now, what is this brassica? Brassica, it is nothing but your mustard. Okay, rapeseed mustard. So this brassica, basically, this is going to be known as pusa swarnim, and this is going to be against white rust. Just like how we had brown rust, same way we are going to have white rust. Okay, then we have cauliflower pusa shubra. Okay, and what is this going to be? See, right now we talked about black rot of cauliflower, right? So black rot and curl blight, black rot. Okay, so basically these are two different varieties. Now I tell you how to remember this thing, guys. See, cauliflower, this is going to be white in color, mostly white. Okay, and if you are a Maharashtrian, generally what do we say? Pandra shubra. So white shubra, pandra shubra. It is this way. Okay, so this is how you will remember it. Okay, then we have your chilies. Okay, so chilies, basically this is going to be known as your pusa sada bahar, and this is going to be against your chili mosaic virus, tobacco mosaic virus, and your leaf curl. Okay, so pusa sada bahar, why? Because it is going to grow throughout the entire year. That is why sada bahar. Okay, guys, these names are very important. Try to remember it this way only: Himgiri, Hillbunt, Brassica, just Pusa Swarnim, White Rust. You have to remember this because this is a little odd one. Pusa Shubra, that is white, Pantra Shubra, and against Black Rot, because we've already understood it out over here that is going to be against Black Rot. And chili, Pusa Sadabahar, grows throughout the entire year. So chili mosaic virus and tobacco mosaic virus. That's liquid. Okay, so guys, keep this thing in mind. Okay, keep this thing in mind. Okay, but now guys, after this, what we did is we did not stop out over here. This is one type of your breeding. Okay, uh, now. Just go through this ones. Go through this ones. Then we'll move on to the next one, that is your mutational breeding. Okay, is it fine now? Okay, done. Okay, so now after this, guys, we are going to move into your mutation breeding. Okay, so let's try to understand what is this mutation breeding. Okay, so guys, see, generally when we talk about mutation, mutation is like, a sudden genetic change that is going to occur inside the plant, right? A sudden genetic change which is going to occur inside the plant or an organism, and that is going to be transmitted from one generation to another generation. That is nothing but your mutation. Okay, so let me just put down what is a mutation. This is nothing but a sudden heritable change in the genetic material. Heritable in the genetic material. Okay, but now, guys, what happens, see, naturally also mutations occur, but it takes a long, long time for mutations to occur. So what do we do is, rather than waiting for, rather than waiting for nature to induce mutation and then we will select that mutation, rather what we go is for artificial mutation. So let's try to see what is this artificial mutation and how we can bring about. Now, guys, the chemicals which bring about mutation, we call them as mutagens. Okay, so mutation generators. Okay, mutation generators. So these bring about or these cause mutation inside the organism. Okay, but now what are you going to see is, you're going to see that these mutagens, they can be of two different types. Okay, now either they can be your natural mutagens or they can be your artificial mutagens. Okay, artificial meaning they are going to be your chemical. Okay, but now, guys, when we talk about natural mutagens, how can we mutate organisms naturally? So how is this generally obtained is, guys, sometimes you are going to use extremely high temperatures. Okay, otherwise you are going to use extremely high concentration of your carbon dioxide. Okay, because carbon dioxide is also going to be deadly for your organisms. In excess of carbon dioxide, they are going to feel as if they are going to be asphyxiating, and then they are going to die off. Okay, so remember, high concentration of carbon dioxide is also going to be really, really bad. But then, along with this, guys, you are going to have your x-rays. These x-rays, they are really, really deadly. They have so much of power that they can even knock off your genes. So we use these x-rays, and the most potent mutagen, most potent, this is UV radiations. Guys, with these UV radiations, you actually get loads and loads of mutations every now and then, because there is something called as thymine dimer formation that way. Okay, then the next thing is, we are going to have chemical mutagen. Now in chemical, basically we are going to make use of chemical agents. So what are we talking about, guys? We are talking about some of the famous one. We have your ETBR. Okay, ETBR is ethylene bromide, basically. Ethidium bromide. Okay, we call this thing as ETBR. Okay, so we have ETBR, then we have EMS, ethyl methyl sulfonate. Okay, this is also a really, really potent one. Okay, I have worked on these things, so so I know these are like really, really deadly stuff. Okay, so we call this thing as EMS. Along with this, we have colchicine. Now, do you guys remember where we had used colchicine? Where we had come across? Anybody? What is the importance of colchicine, guys? Yes, very nice, Neha. This is going to arrest your mitosis. But can you tell me? It is going to prevent something. What is it going to prevent? Very nice. It is going to prevent spindle formation. Okay, and lastly we have mustard gas. Okay, so these are some of the chemical agents, okay, that are going to be used to create mutations. Now sometimes, okay, sometimes when we have seeds and all, okay, so these are like, cells, what we are talking about. But if you want to bring about mutations in seeds or seedlings, like, small, so you are going to see that seeds or sometimes seedlings. Yes, 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 yes. Okay, you will see that these seeds or seedlings, guys, they are sometimes, uh, you know, exposed to cobalt 60. Guys, cobalt 60, this is like, really, really deadly one. Okay, cobalt 60. Or we can make use of your UV or your X-ray machines. Okay, especially we have an experiment where we'll be talking about X-rays on use of X-rays on fungal spores. So etc etc etc. So this is generally done with regards to your seeds or seedlings. Okay, but now the next thing that is going to be is, guys, what are we going to do? Okay, now let's imagine what is the plan. How do we get these disease resistant plants? So what we do is, we are going to take a plant basically, or we're going to take a plant cell, or we're going to take a seed or seedling. Okay, so let's say we have your plant cell, or we have your seed, or we take your seedling. Now what are we going to do is, we are going to expose them to mutagens. Okay, now once we have exposed them to mutagens, then what? Mutagens are going to cause some kind of your problem or mutations inside the cell. Then we are going to take these, and then we are going to screen them. Like, you're going to, if it is a seed, you're going to sow the seeds and you're going to see. Or if you're having a seedling, then you're going to sow the seedling and then you're going to see how is it going to react to different kinds of your diseases or your pest as such. So after this, what are we going to do? We are going to have screening. Okay, this is screening, okay, to test for disease resistance or for pest resistance. Okay, it can be this, or we can also look out for your increased yield, etc. Okay, so this is generally how your mutational this thing is done. But guys, what are the examples? Like, how have we got different kinds of your pest resistant or your disease resistant plants? Okay, so let's try to see how we have got through mutational breeding. So the first one, guys, we are going to talk about is your moong beans, basically. Okay, so we have these moong this thing, which are, I'll just, one minute. Right, so examples that we have got out over here is, first we have developed moong beans, and these moong beans, they are going to be resistant to, okay, so they are going to be resistant to moong mosaic virus. Okay, so resistant to moong mosaic virus. Or the second one is powdery mildew. Okay, moong mosaic virus and powdery mildew. In powdery mildew, what happens once again, it is just like your rust kind of thing. So these beans and all, they immediately start, showing pitting, and then they start, being powdery, flaky kind of thing. That is what is going to happen, guys. Okay, the next one that is out over here, guys, we are going to talk about your Jagannath rice. Okay, so another one we have. Jagannath variety of your rice. Okay, Jagannath variety of rice. Then we are going to have like a Regina 2 variety of cabbage. Okay, Regina 2. This is of cabbage. Guys, this Regina 2 basically, this is resistant to your bacterial rot. Okay, so against, okay, resistant to, this is going to be your bacterial rot. Okay, then after that, we are going to have your indoor 2. Indoor 2, this is going to be of your cotton. Guys, always remember cotton is going to be like susceptible to cotton bollworms. Okay, so this is going to be against your bollworm. Okay, we will always target this thing, the bollworms. Ball is what cotton ball that we get. Okay, and when it is infested by your worms, we call it as bollworms basically. Okay, also there is another one that is your NP. NP836. This is a variety of your wheat, and this is resistant to your brown rust. Okay, so this is going to be your against your rust. So that way, guys. So this is how we are going to have some of these. Okay, some of the plants which have been obtained by mutation breeding. Okay, once again, for your mutations, always remember we are going to incorporate your mutagens that way. Okay, but now after this, okay, so guys, just have a look at this. Just see if anybody has any doubts. Jagannath, this is also resistant to your rust. Okay, this is also resistant to your rust. Sir, how do they exposed to cobalt 60 as it is element? So it's the same thing. See how you have a nuclear reactor or how you have a radioactive reactor. It's the same thing. This cobalt 60 crystals, they are going to be like, stored inside, let's say, an enclosure. You are going to take your cells, you're going to put it into that enclosure. They are going to be kept in that enclosure for a few minutes, or like, depending upon what dosage you want to give or how much like time it is going to be, and then you are going to just put it inside and then you're going to remove it out. That is how you expose it. Sir, cabbage is what? Cabbage is your largest apical bud. But I don't know, like, why are you asking this? Are you confused between cauliflower? Cauliflower is apna flower, flower, white flower, basically. And cabbage is nothing but patta gobi. Some people call it patta gobi. Bacterial rot. This is against your bacterial rot. Acha, acha, acha. Okay, 1 minute. No, bacterial rot. Sir. Okay, chalo. So, guys, now let's try to move ahead and let's try to see the next thing, guys. Okay. So the next thing is, guys, just like we have, like, we developed like plants for some kind of your this thing, like is like some kind of your disease resistance. Same way, we can also develop the same thing for your pest resistance. Okay, so let's try to see for your pest resistance, what are we really going to do? Okay, chal. Okay, so for pest resistance, guys, who are we really going to talk about? Now, guys, what happens is, now always remember, okay, whenever we talk about like plants, okay, plants, they are generally going to be grazed upon by your grazing animals, or they are going to be like, eaten by like, the just the passerby animals and all. Okay, so some of them, they are going to develop different kinds of mechanisms in order to avoid them being from eaten. So for example, we are going to see thorn formation. For example, sometimes we are going to see your the hair like structures formation. Okay, so they're going to be doing two things. It is first going to offer defense to the plant, and at the same time, you are going to see that it is also going to prevent the pest from setting on top of it, or they are going to develop certain compounds because of which, if by any chance the pest they are going to eat it or something, the pests are going to die. So that is the idea between developing your pest resistance in plant. So, like, what is going to happen? Let's try to see. Okay, so guys, in this case, we are going to develop plants. Okay, once again, it is going to be by your normal breeding only, and we are going to screen for these characters. So, what are we going to see is, guys, you are going to see if by any chance we are having hairy leaves, okay, really, really hairy leaves in cotton. This is generally seen in the case of cotton. Now, what is this cotton? And the other one is your wheat variety. Okay, so if it is going to be there in your cotton and wheat, what is this going to be resistance again? Guys, this is going to provide resistance against Jacets. Okay, I'll just put down. It is going to give resistance against jacids or jacids. Once again, these are like small insects, like uh, small insects kind of stuff, things basically, like your like ticks kind of things basically. Okay, then next one is like they are also going to take care of your beetles. Okay, so we have different kinds of beetles, but they're going to act on that as well. Okay, etc, etc, etc. Okay, so this is one variety. Second thing, guys, sometimes the plants, they are going to have like really, really solid stem instead of like having a really hollow stem. That is another variety. So what is going to happen in this is, you are going to see that stem, this is going to become completely solid instead of your hollow stem. Where will we see this thing, guys? This is seen in the case of your wheat. And what is it going to give resistance against? This is going to be against your stem borers. Okay, so see, we had already talked about like corn borers, or we were having your ballworm basically borers. So like that, we have your stem borers. Okay. Now, another thing that we're going to see is, guys, you are going to see sometimes certain compounds, they are going to be like really, really important. Their concentration will depend on like, how it is, how the plant is going to react to different kinds of your pests. So, for example, guys, when we talk about your maize plant, okay, so in your maize, what happens is you are going to have extremely high levels of aspartic acid. Okay, so we have aspartic acid, which is going to be high levels, and you are going to see a little low levels of your nitrogen. So nitrogen levels, they are going to be low, and sugar concentration, sugar content, even this is going to be low. If we have this combination in the case of your maize plant, guys, then this is going to give you resistance against your stem and fruit borers. Okay, so once again, this is going to be against your stem borers or your fruit worms basically. Okay, Fine. Next thing, guys, you are also going to have another variety. Now, most of the times, what happens is in those cases, guys, in those cases where the plant is producing nectar, in that case, all the insects are not going to be attracted towards it. In that case, what happens is, guys, the plants they stop producing nectar. So you will see you are going to have nectarless, okay, nectarless plants, plus you are going to have extremely smooth leaves. Now because of this, what is going to happen is these pests and all, they cannot easily climb on, or they cannot easily hang on to your leaves, okay, so they fall down again and again. This combination, where are we going to see this thing, guys? This is going to be seen in your cotton, okay. And now because of this, guys, what is this going to give us protection against? This is going to give us protection against your bollworms, okay. Always remember, guys, whenever we talk about your cotton, cotton is always going to try to have some kind of resistance against your bollworms only. Okay, but now, guys, we have another one. Okay, we have another who are going to be very important. So, for example, we are going to have bhindi, we are going to have rapeseed mustard, we are going to have your beans. So, guys, let me just take this thing and let me just show it to you. Who are the other ones that we need to know? Okay, so guys, we have three of these, okay. Apart from the ones that we have listed, we have three of this. So first one, we have brassica. This is nothing but your rapeseed mustard, okay. What is this? This is known as your pusa gauro, okay. And guys, this is very important, okay. They will always ask you about pusa gauro, okay. What is this going to be against? It is going to be against your aphids, okay. Chalo. Second one, we have your flat beans. Pusa Sem 2, Pusa Sem 3. Okay, once again, jacids, aphids, and fruit borers. You just have to add all the problems out over here. And finally, we have your okra. This is Pusa Sauni. Earlier, guys, we had seen Pusa Swarmine. Okay, just see out over here. That was another one. Pusa Swarnim. Okay, this was another one. And Pusa Sauni, this is another one. Okay, guys, don't get confused. Pusa Sauni and Pusa A4. Once again, why are they against? They are going to be against your shoot and your fruit borers. So this is what we need to know with regards to your pest resistant varieties of your plants. So guys, just have a look out over here, just see if you are okay. Then we will move ahead with the next one, guys. Okay, chalo. So, guys, done everyone. Let's try to move ahead. Guys, these names are very important. Okay, somehow or the other, you just need to remember this thing. What is Pusa Gauro, Pusa Sauni, Pusa Swarnim, Pusa A4, Pusa Sem 2, Pusa Sem 3? You just need to remember this thing some or the other way, guys. Okay. Okay. Chalo. Let's try to say the next thing, guys. Okay, now the next thing that we are going to go is, see, we are doing like, we are getting plants for like, different kinds of purposes. Same way, we also get plants for increased, increased nutritional value as well. So let's try to see what is this thing exactly. So next is like, plants for improved varieties, improved food quality. Okay. So let's try to see how we will go about this thing. Okay, so guys, we are going to talk about something that is called as your biofortification. So let me first make you understand what is the difference between fortification, and then we'll talk about biofortification. Okay, so guys, let's try to see what is this. Okay, so for this thing, guys, we will generally go for a method which is called as your biofortification. Now, what is this biofortification? Guys, generally remember, okay, what happens is, okay, how the processing is done. Let's say we have maize. Okay, let's imagine we are talking about maize. Now, maize initially, it may not be like, that nutritious. Okay, so what people do is like what companies do at the time of processing, they will process your maize. Okay, let's say maize flour, corn flour, or maybe maize products and all, but then to increase its nutritional content at that time, at the time of processing, what they do is they add extra minerals, or they add extra vitamins, or they add some extra stuff. When you are going to add extra stuff at the time of processing of the product, then it is called as fortification. Okay, so you must have heard of fortified atta, fortified fortified poha, fortified salts sometimes, right? So iodized salt is fortified salt. Okay, so that is nothing but your fortification. But now, when we talk about biofortification, what does this mean? Biofortification means you are going to improve the quality of the plant itself. So while the plant is growing, you are going to give it so much amounts of nutritious food or nutritious fodder, so that it is going to develop the nutrients inside itself only. That method is called as biofortification. Okay, so this is one. But guys, by biofortification, what are we really trying to do? Again, what is the aim of the entire thing? So basically, by biofortification, by increasing its the quality at the source level itself, the aim is to increase either your vitamin content. Okay, so either we are trying to do this thing, or we are going to try to like improve the oil content. Okay, so vitamin content, or it can be your oil content, it can be your nutrient content, micronutrients, or whether it is your macronutrient. So even that can be increased by this thing. Or for example, we can also increase your protein content. So even that can be increased by this method. Okay. So once again, biofortification, I told you, you are going to increase the quality of the plant while it is growing only. And this biofortification can be achieved by different things. It can be achieved by a normal breeding practices also, or else you can also take like the help of your normal new, okay, the new biological techniques, that is your recombinant DNA technology. So even that is possible. So I'll just bring this thing. This is going to be brought about. Okay, so one, we can have these selective breeding practices, or we can use this rDNA technology, that is your recombinant DNA technology. Okay, so by doing this, we can achieve our aim. But then, guys, what are some of the examples? Okay, so let's try to see some examples out over here. And guys, you need to remember this thing. Okay, both important from your boards also and from your neat point of view as well. So, guys, the first one that we have something that is called as your hybrid maize. So this is going to be some fortified maize. But then what is the importance of this thing, guys? What we will observe is this is the one which is going to have twice the amount, increased production, absolutely high production. So this is going to have twice the amount of lysine and tryptophan. Okay, these lysine and tryptophan, these are nothing but your amino acids. Okay, so they are going to have extremely huge amounts of these things. Okay, another one, the one which was like very very important in our green revolution, that is nothing but Atlas 66. Okay, so Atlas 66, this is nothing but this is your wheat. And why is this important, guys? This is going to be your improved or high varieties or high quantity of your protein. Okay, third, just like this, we have iron fortified rice. Okay, and this is going to have around 5 times more iron. Okay, so really, really rich in your iron. Okay, next one, guys. After this, we are going to have another one like carrots. Okay, so when we talk about carrots, etc., you are going to see that they are going to be built in such a way that they are going to have increased vitamin A. Already they are rich in vitamin A, but still you are going to increase its vitamin content. Also, the next one, we have your tomato, or we have your bitter gourd. Okay, now what is this bitter gourd? Can anybody tell me bitter gourd? We have normal gourd and we have bitter gourd. What is this bitter gourd? Very nice. Yeah, this is nothing but your Karela. Okay, so in this Karela and tomato, basically they are trying to increase your vitamin C content. So enriched vitamin C content. Okay, so these are some of the plants that have been developed by your this thing biofortification. Okay, so guys, once again, just have a look out over here. Then we have one more type of modification. Okay, guys, let's move ahead. Everybody, all right? Okay, now, guys, just like this thing, 5 times. It is 5 times more. This is 5 times. Okay, guys, now the last type of modification. Okay, and now remember, this is called as your genetically modified plants. Okay, or genetically modified organisms. We call them as GMOs. Okay, so we have lasting genetic modifications. Okay, so somehow or the other, guys, by using your technologies or your biotechnological tools and all, we can now create new plants altogether. Okay, by making use of plasmids, by making use of the desired genes, then we are going to convert it into your recombinant DNA. Then we are going to put it somehow into your plant. Okay, but guys, by this genetic modification, which are the plants that we are going to give rise to? So guys, the most important example out over here, that is of golden rice. Once again, this golden rice was the most important thing. Just like we have Atlas 66 golden rice, this helped to overcome, like the nutritional qualities of people masses, and plus it helped in your green revolution. Okay, so we call this thing golden rice as your GMO, genetically modified organism. Okay, now what happens is, this basically, in golden rice, how have we prepared this thing? Okay, so basically we have taken genes from two different things. Okay, so let me just say, it has genes from one, it is going to be agrobacterium. Sorry, it is from your soil bacterium, that is your Erwinia. Okay, so basically this is first thing, it is going to be from your soil bacterium, Erwinia. Okay, and the second one, guys, the second genes we are going to obtain it either from your daffodils, or we are going to obtain it from your maize. Okay, either of the two. And then, what is the best part of this, guys? Whenever we have this combination and when we get this golden rice, guys, this golden rice has really, really high amount. It has high amounts of beta carotene. Okay, and guys, what is this beta carotene? Beta carotene is nothing but this is a precursor of vitamin A. It is a precursor of vitamin A. So when you have this thing, guys, what is going to happen? This is going to enter into your body as beta carotene inside your body. Immediately, this is going to be converted to your vitamin A, and this is how it is going to take care of your vitamin A deficiencies, guys, so in the body. That's it. Carrots have beta carotene, but not exactly vitamin A. Yes, carrots have a lot of these things. Okay, so basically, carrots have carotene as well. Especially beta carotene is like where in your beetroot. Those are really rich in your beta carotene. It is that way. So basically, once again, when we talk about carrots, they have carotene, but then once again, you need to convert it into vitamin A as such. That. Okay, so finally, this is the last one out of the modifications, guys. So with this, we have completed all the modifications. After this, guys, we are going to have the next thing, that is your tissue culture. Like, what exactly is tissue culture? And finally, we have one last part, that is your single cell proteins. So that's it. And with this, then we will be able to finish off with your chapter. Okay, so guys, what we'll do is, once again, we'll break over here for 5 minutes. Okay, so does eating carrots really help for eyes? Yes, absolutely. It does help. Why? Because see, carrots have a retinol. Sorry, carrots have your this thing. Okay, retinol. Okay, that is nothing but it is going to be vitamin A basically. Okay. So what happens is, when you have carrots, okay, the vitamin A content which is there in the body, that retinol is going to be used for proper functioning of your retina. That is why it is going to be very very important. So because we have the rods and cones, guys, so those need some, these vitamins and all. So that is why, having carrots is going to be like a really really important thing. Same goes with beetroot. What is besides maize? Daffodils. Daffodils. Okay, beetroot is really, really rich in your vitamin A, guys, meaning your beta carotene. And that beta carotene will, if high quantities of beta carotene will immediately convert into vitamin A in your body. So yes, having beetroots is going to be really, really important thing. In fact, if you know something, if a person is suffering from low blood levels and all, people will actually ask them to eat beetroot, and they'll also ask them to have khajur. Okay, so it is the same thing. Both of them, they help in like increase in your blood concentration also, and beetroot also in maintaining your vitamin A. Yes, it has iron also, right? Okay, chalo. So guys, what we'll do is we just break over here quickly for 5 minutes, and then when we come back, we'll start talking about what is your tissue culture. Okay, chalo. So, guys, let's get back. So, the next thing we're going to talk about is what is tissue culture? How is it going to be like, and what do we really need to know? So, first, what I'll do is I'll give you an idea about the requirements. Then I'll walk you through the entire process of tissue culture, how we are going to do that entire experiment, and then we'll see a few details, what is the medium, how is it going to be, what are the different components of the experiment, what should be done, and all. Okay, that will be seen a little later. First, let me just introduce you first to the requirements and second to how the experiment is going to be done. Okay, so guys, tissue culture basically this is like a rapidly developing technology by which, guys, we can actually create large number of plants. Okay, a really large number of plants, even from one single cell. That is nothing but tissue culture, and that is why this is very important technique. Okay, so guys, let's try to see what are the requirements first. So first thing, guys, that we're going to require for this is called as an explant. Now, explant is what, guys? Explant is nothing but it is your plant tissue only. This is a plant tissue which has been removed from the plant. Okay, that is called as your explant. So first is explant. Second thing, guys, we need a medium. See, just like how we have a medium, for growing bacteria and all, right? Same way, we require a medium to grow all these cells. So second one is going to be our medium. Okay, this is known as your MS medium, that is your Murashige and Skug medium, or there is another medium also. We'll talk about the NOPS medium. So MS or NOPS medium. Okay, so we'll talk about this a little later, but these are the two media that we'll be using. Okay, third thing, we require aseptic conditions. Now, aseptic conditions meaning germ-free conditions. Okay, so that now there is no contamination, okay, of our cells while we are working. So aseptic condition, that is your contamination free. Contamination free environment. Okay, next thing, guys, we require equipment. Okay, so now equipment, what to check, whether the temperature is proper, okay, the whether the mixing is proper, whether the vortex is proper, whether the mix the maintenance of your media is proper. So for all these things we require different kinds of equipment. So equipment to check conditions. Okay. And finally, guys, we are going to require some plant hormones. Especially, we have your auxins and we have your cytokines. Okay. Now, guys, by making use of different combinations of your auxins and cytokines, you can initiate either your stem generation, collagenesis, or you can initiate your root generation, that is rhizogenesis. So these two, they are going to be very important, guys. Okay, so very important. Okay, but now first let's try to see how is this experiment actually done. What really is happening out over there. Sorry. Okay, so the experiment is very simple, guys. Okay, so nothing to really worry as such. Okay, so first let's try to understand what is happening. Then we'll slowly talk about some details of the requirement. So, guys, let's imagine. Okay, let's imagine we have this plant. Okay, now let's imagine this is the last plant standing on the earth. Let's imagine. Okay, so one of its own kind, one of its variety. Okay, this is the last of the varieties which is standing on top of this earth. Okay, but now what happens, guys? Suddenly, let's say this plant gets infected. Okay, oh my god. So if this plant is infected, it is going to be bound to die. Now, what do? We as scientists, what are we going to do? We take this apical meristem. Okay, now always remember, guys, we are going to go for your apical meristem. Why? Because even if the entire plant is diseased or it has got infection, guys, the apical meristem will never be infected. It is going to be still remaining free from the disease, and that is why we can use that. Okay, now, guys, what are we going to do? Okay, you have taken this. Now let's say we are going to cut this part. Okay, So if we have cut this part, guys, what is this going to act as? This is nothing but this is going to be our explant. Okay, now I need to use this thing properly. So guys, let's say we have got our explant, and this explant is going to contain rapidly dividing cells, and plus they are going to have their own properties, meaning they have their own genetics, they have their own genome, and which is what we are trying to solve. So we have cut the apical meristem. The apical meristem is going to look something like this. Now see all these cells which are there out over here, they are going to be like lumped up. They are going to be cramped up one besides the other. Now I want these cells to separate. I want these cells to multiply. So now what are we going to do, guys? We are going to take a medium. Medium is going to act as what? It is nothing but it is going to be food for your bacteria, food for your the explant. So now what are you going to do, guys? You are going to take this explant and you are going to put it out over here into this medium. Now, initially, when you put this thing, guys, okay, it is going to be something of this sort. Let's imagine you are stirring this. Okay, now because you are stirring this, guys, is this entire blob of or mass of cells going to withstand that thing? No. Immediately you are going to see that this starts dispersing here and there inside the medium like this. Okay, so now immediately it starts growing into this medium. Now what happens, guys? As it starts growing, now you're going to see that, oh, it starts multiplying. So they are going to start multiplying like this. As they're going to start multiplying, guys, you are going to see that, oh, they are going to be like individual cells, but now they have like started growing up. Okay, now what? In the next case, guys, what am I going to do? I know for sure now these cells are ready to give rise to a new plant altogether. Why? Because these meristematic cells, guys, they have the property of totipotency. What do we mean by totipotency? Any cell which is capable of giving rise to an entire organism, that is going to be your totipotent cell. Okay, so now what are we going to do? We are going to take one drop. See, guys, we even if we take one drop, there are going to be millions and millions of cells out over there in one drop. So what we do is we are going to take the same medium what we used for food out over here. We are going to make use of the same medium, but rather this time we are going to solidify that medium. Okay, so this is solid medium. Okay, in order to avoid any kind of contamination, you're always going to plug these test tubes from the top like this. Okay, now in the meanwhile, guys, what had you done? You had taken one drop. Okay, so with the help of a pipette or with the help of a dropper, you can just remove one drop. And that one drop, guys, it is going to be transferred onto this slant like this. Okay, but now immediately what is going to happen as you add this drop, all the water and all everything is going to be absorbed, and now you will see only the cells being present out over there. Now what happens, guys? Let's fast forward a few weeks. Okay, immediately, and plus this also depends upon like what kind of a plant you are having, but generally in a week or a couple of weeks you are going to see tremendous results. So now what happens, guys, is you are going to once again leave the test tube as it is. Okay, and now what you will see is that on this plant, this small plant, it slowly it starts growing like this. Okay, so basically this is ultimately going to give rise to a plant, but before it gives rise to a plant, guys, you are going to see that this small mass basically this is going to develop like it is going to show you like some small development out over here. It becomes a mass like structure. What is this mass like structure called? This is called as our callus, right? This is known as callus, and this callus, guys, later on it is going to develop into a new plant altogether. So slowly you have a new plant something, a new plantling or a seedling kind of thing, like this. And now once we have got this thing, see, from one cell we got, like, one cell or one drop, we got one plant. Like this, there are going to be so many milliliters of your medium out over there, guys. You can just take this thing and you can cultivate more and more and more and more plants. Okay, so guys, this is our entire tissue culture technique. Okay, everybody is okay with this thing? First, go through this, just see if you have understood this part. The same thing we are going to do in suspension culture also. I will talk about what is suspension culture. Okay, everybody all right? Anybody has any doubts from here? Okay, guys, always remember, in your exams they ask you what is callus. Okay, callus is nothing but undifferentiated mass of cells. Okay, and in this medium, guys, okay, whenever we are talking about this medium, in this medium you are also going to add your auxin plus cytokinin ratio. Okay, so depending upon what you want first to come out, like, where should maximum growth be concentrated, so for that. Okay, so any doubts, anybody has any doubts, anything you wish to ask? Okay, chalo. So now, guys, let's try to move ahead and let's try to see how do we really do this experiment. Like, what are the different things that are going to happen out over here? Okay, so first of all, guys, what property of the plant are we like tackling out over here? I told you we are tackling your totipotency. Okay, there are like two types of cells. One is totipotent cell, pluripotent cell, but more or less the meaning remains the same. Okay, so we are talking about totipotency. This is nothing but ability of the cells to give rise to an entire organism. Okay, so this is totipotency. Guys, first thing, what are we going to go for? We said we are going to have our explant. Guys, this explant generally, okay, generally we are going to go for your apical meristem. Okay, you can take others also, but this is always going to be disease free. So you go generally for your apical meristem. Okay, but now, guys, when we talk about this, the experiment, nature of this entire explant, how are we going to, like, get this thing going? Okay, so guys, first thing we said, we have this explant and we need this medium. Okay, so what is this nutritive medium? Okay, so I'll just mention out over here, what is this nutritive medium that we are going to use. So guys, this nutritive medium which is there, generally what we do is we maintain the pH to be in the range of 5 to 5.8. Right? This is considered to be a good pH for the seedlings to grow as well. Okay, meaning the small plants, the plantlet that way. Okay, and this, this entire thing, this nutritive medium, along with your hormones. Okay, so plus hormones, that is your auxin and cytokines. Okay, you are going to see that this nutritive medium could be of two types. Now, generally, if it is liquid, we call this thing as broth. Okay, and which are these? Which is this medium that we are going to use? Either we are going to use Knops medium. Now there are two. Okay, one is Knops medium and one is Murashige and Skoog. Sorry. Okay, so we have NOS medium. And generally, guys, when we talk about NOS medium, what are the main important components of this? you are going to have salt, and you are going to have excess of your sugar. Okay, so that is going to be one type of your medium. But now the other one, guys, I want this liquid medium to convert into solid, because see here it was liquid, but here it was all solid out over there. How will I convert this liquid medium into solid medium? So solid medium, what we do is we take the liquid medium and plus we add agar. So then it becomes solid. Okay, so plus agar. Now, why do we generally use agar? This agar, it solidifies the medium. That is why. Okay. Okay. And now, guys, which medium are we talking about? We are talking about the new MS medium, that is your Murashige and Sku. Okay. So, first thing is that. Okay. But now, guys, when we talk about the explant, okay, what is the explant that we are going to really take? Okay, so once again, when we talk about the explant can be of three different types. Okay, so you can either use your cells, okay, how we had basically, and then we call this thing as your cell culture, basically. Okay, if you're using cells, then we call this thing as cell culture. Second thing, if you're using organs, altogether organs, then this is known as your organ culture. And the third thing, guys, we can also help your embryo. So then that is called as your embryonic culture. Okay, so embryo culture, that way. So depending upon explant, we can have like three different things. Okay, everybody all right with this thing? Fine. But now, depending upon the growth, like how the growth is obtained. Okay, so we have like two types. Either we have your solid culture, or we have your suspension culture. Okay, so let's try to see what is the difference. Just a second, I'm getting a call from the classes. Oh, 1 minute, 1 minute, 1 minute. Okay, sorry. Okay, so guys, based on how we are growing this thing, we are going to have two types. Either we are going to have your callus culture, that is your solid medium. So what we had done, see guys, here we were doing callus culture, okay, because we produce this callus. Okay, so we can do this thing as callus culture. If you're doing callus culture, this is going to be on solid medium. Or the other way around, guys, we also have this as your suspension culture, which is going to be done in your liquid medium. Okay, but ultimately, even in this suspension culture, ultimately we need to put it onto the medium only, like liquid medium. But I'll explain you how, as we move ahead. Okay, chalo. So this is the first thing that we need to know about your explant and your nutritive medium. But guys, you know what? Whenever we work with these plant things and all, or whether it is your microorganisms, there is always a chance of contamination. Contamination from other microorganisms, contamination from bacteria, contamination from environment. It can be anything. So I cannot do this thing openly, like how we do, how we cook food in our kitchen. I cannot do this thing, so I have to do these experiments in your aseptic condition. Aseptic condition meaning contamination free environment. There should be no bacteria, no fungus, no virus, nothing. It should be completely free of this thing, of your uh, completely free of your germs. So guys, how do we maintain these aseptic conditions? Let's try to see. Okay, so when we talk about this aseptic conditions, guys, we call this process as sterilization. And this sterilization, it can be of two types, guys. It can be either your wet or it can be dry. Okay, so first of all, what is this wet? Now, wet sterilization, guys, this is generally done for your explant. Okay, because you are going to remove the part of the plant and then you have to wash it or to rinse it off through so that all the germs and all they are they are removed. So guys, how do we do this thing? So either you can take 70% of your alcohol and you can rub it on the surface of the leaf. You can use the same thing, that is 70% of your alcohol, use it against all the instruments that you're going to use. Okay, second thing, either this or else we can use 1% sodium hypochlorite. This is a known, okay, a very very common known disinfectant kind of thing, okay, antiseptic solution. Okay, so sodium hypochlorite. Okay, this is even, in fact, if you go to the dentist and all, they will use this sodium hypochlorite only. Okay, or else we have 10% of your H2O2, hydrogen peroxide. So we can use this as well. Guys, if you still don't want to use this thing, we still have another. Like, sometimes you can use your HgCl2 as well. Like, 1%, 0.1% of HgCl2. This is your mercury chloride, basically. Okay, so guys, we can use this thing now. If mercury chloride, guys, this is really, really deadly. Okay, none of the enzymes work properly in the presence of this mercury chloride. Okay, but now, guys, this is for your wet sterilization. But for your dry sterilization, what are we going to do? For dry sterilization, guys, we are going to use, sorry, we are going to make use of your like a, a basically it is going to be like your cooker. Okay, how we have cooker at our places, like it is going to increase the temperature, it is going to increase the pressure. Okay, like that industrial cooker. Okay, when we talk about industrial cooker, that is called as autoclave. It is huge in size, so that it can incorporate large amount of the medium utensils, your vessel, your like your glass pipettes, okay, your beakers, whatever you require for doing this experiment, that can be incorporated into that. Okay, so this is generally okay. So this dry one, guys, we are talking about is your autoclave, and especially in your autoclave, guys, what are we going to see? You are going to have high temperature. Okay, so you're going to encounter high temperature, you are going to have increased pressure, more temperature, more pressure. Okay, and this is going to be used for mostly your glass equipment. Okay, glass equipment. media, etc., to sterilize the media. Okay, so this is the second thing, guys. Okay, but then after that, we have talked about your this thing, the okay. Okay, after this, we have talked about your auxins and cytokines. Now, guys, remember, depending upon what the ratio is, how you're going to use that ratio, there are going to be two things. Generally, auxins, if they are going to be more, always remember, auxins are responsible for root formation. Auxins, they will help in root formation. And this root formation, we call this thing as a rhizogenesis. Okay, this is known as rhizogenesis. Second thing, we have cytokines. Okay, cytokines, this is going to be for your shoot formation. And what is this? This is called as your collagenesis. So rhizogenesis and we have collagenesis. Okay, now guys, okay, when we talk about this entire medium, okay, always remember, okay, all your this thing, like before experiment, I will just mention, okay, I'll just explain. Okay, before your experiment, what we have to do? Okay, we have a UV chamber. Okay, so basically, I'll just give you an idea. Okay, what we do is we have this kind of a unit. Okay, this is called as a laminar airflow. You have a desk like thing out over here on which you can keep all your things. Okay, this place is like to sit. Basically, you can keep your chair out over here, or you can keep your stool and you can sit on top out over here and you can work like this. Okay, you work out over here this way. So what we do is, guys, this in your laminar airflow, we equip this with a UV tube light. Now see, I told you UV is a very potent mutagen. So what we do is this UV tube light, we are going to switch it on. Okay, now see, you are not going to sit out over here anymore. Okay, because otherwise you will also face mutations. Whatever things that you are going to use, like whatever instruments, whatever equipment, whatever the media and all, what we do is you are going to take this, you are going to put it like so. Let's say I'm keeping some flask out over here, let's say I'm having some test tubes out over here which you're going to require, okay, while doing the experiment. So you take this thing, you're going to have pipettes out over here, and then you are going to have this burner as well. Okay, this burner helps in like uh, dipping your things in fire, I mean dipping your things in alcohol and then just firing it off, okay, to remove your entire bacteria that way. So you keep this thing and you switch on this thing for minimum like 15 to 20 minutes. Okay, your textbook says 1 hour, that is not possible. Okay, so you keep, see, you're not keeping your cells. Okay, you cannot keep the cells, otherwise they will be mutated. So this UV tube light, okay, you are going to switch on. Okay, going by your textbook, it is going to be 1 hour. Actually, it is not 1 hour. It is going to be just 15-20 minutes. Okay, that is it. Okay, 1 hour to sterilize equipment, media containers, etc. Okay, etc., etc. So all these equipment, you are going to keep it in this thing. You switch this thing on for 1 hour, and after 1 hour, then switch off, and then you can start with your experiment. Switch off and start experiment. This is how this laminar airflow really works. And guys, what happens is this laminar airflow, this maintains a septic environment because there are like different kinds of filters and all fitted inside of it. There is your airflow out over there, Okay, it will make sure that 99.99% of germs, bacteria, whatever they are going to be removed from the this environment where you are working. Okay, so maintains a septic environment. Okay, but now guys, let's say when we start doing this experiment, okay, after we do this experiment, guys, what is the pH of the medium that we are going to use? The pH of the medium, I already told you, it is going to be in the range of let's say here, pH in the range of 5 to 5.8. Okay, so the same range we are going to use out over here. So pH of the medium, this is going to be roughly in the range of 5 to 5.8. And guys, the temperature of the medium, we need to maintain the temperature throughout. Okay, this is going to be around 18 to 20 degrees Celsius, that way. Okay, so you need to keep this thing in mind because this is the place at which everything is going to start. Okay, fine. Okay, next thing, guys, as I told you, we can do this thing in two ways. Either we can go for your callus culture. So this, whatever we have shown, this is callus culture. Then there is another one, that is suspension culture, in which what we have done out of way, we are growing this thing in your suspension. That is your suspension culture. Okay, so callus culture can be directly taken from this thing. So this step can be avoided. In your callus culture, you can directly take an explant and you can just put it out over here and then it will grow on its own. But generally we don't do that. We do it this way. We are going to remove the explant, put it into this liquid medium, shake it for 24 hours or so. Then you will see these cells growing. Then you're going to take this and put it directly onto your solid medium. This is generally actually what we do. And this is when it grows out over here in this thing, that is your suspension culture. Okay, so last thing, I'll just put it down. Okay, so culture methods. So we have one callus culture and we have suspension culture. Only difference is, okay, only difference is you are going to see that this is on a solid medium generally, and here this is going to be made. I mean, they are going to be taking care of this in your liquid medium. Okay, that is the only difference. Okay, now guys, always remember whenever we talk about whether this is callus medium or whether this is just suspension medium, okay, at the end of the day, guys, both of them, they are going to die. So I just cannot do this once and then I will say ki chalo jane do, ho gaya hai abhi, and now I will not require these things properly. No, 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 no. What you have to do is, after a stipulated period, okay, after a stipulated period, guys, you have to take some cells and you always need to inoculate into another test tube and let it grow in another test tube. And every few months you have to do this thing, this. Why? Because this helps in maintaining the purity of the culture. That is why we are going to go for this thing, guys. Okay, everybody all right with this thing? Sure, no problem. Okay, guys, once again, go through this. Just see if everybody is okay. Then we'll move ahead with the next part. Okay, if anybody has any doubts, just ask quickly. Okay. Chalo, let's move ahead. Okay, guys, in your exams, okay, see all these things now. This is like just extra information. What you need to know is you need to remember this experiment. How did we do this thing? And that is it. You have to just describe that, and your job of your tissue culture is done. Okay, so that way. Okay, so guys, this thing, whatever we have done right now, see, we've said that from this explant we got small plants. The same process is also called as your micro propagation. Okay, so guys, let's try to see a little bit of what are the applications of this tissue culture. Why is this thing going to be important? So guys, the first thing that we're going to see is it is going to help in micro propagation, meaning from one entire cell you are going to get an entire plant, and not an entire plant, you are going to get hundreds and hundreds of plants altogether. So this thing is called as your micro propagation. Okay, so first thing is that. Second thing, guys, you are always going to produce disease free plants. Okay, why disease free plants? Because see, you are using your apical meristem, and apical meristem always is going to be free from your infections. So you will always produce disease free plants. Okay, so this, keep this thing in mind. Okay, third thing. Okay, third thing, guys, we are going to see that we produce various types of your secondary metabolites. Okay, from these kinds of your plants. So let's try to see what are these different kinds of metabolites and all. So once again, guys, very important thing. Okay, very important thing. Okay, so guys, we have different plants and their products. So first one, guys, we have Catharanthus. Okay, so Catharanthus roseus. Okay, so this is the first plant. Guys, what is this going to give us? Very important. This is going to give us vincystine. Okay, vincystine and sorry, vincystine. Okay, and vinblastine. 1 minute. Vincystine. Vincystine and vinblastine. Okay, second thing, guys, we are going to have Daucus carota. Okay, now guys, what is this Daucus carota? Daucus carota is nothing but your carrot. Okay, so we have Daucus carota. This is going to give us anthocyanin. Okay, third thing, we have Daucus stromonium. Okay, this is going to give us your tropane. Okay, guys, you need to remember these names. Huh? Then we have Mentha preparata. This is very simple. Mentha preparata, that is going to give us menthol. Okay, and lastly, guys, we are going to have Nicotiana tabacum. Okay, this is going to give us a nicotine. Okay, so these are some of the products, the secondary metabolite things that we have got from here. Okay, next thing is, guys, when we talk about your this thing, that is your micropropagation or your tissue culture, always remember we are going to get large number of plants. Okay, large number of plants. Okay, next thing, guys, you are going to make the plant season free, because see, you can do this thing in the laboratory all throughout the year. So we generate season free plants from this thing. Okay, along with this, guys, okay, endangered species or rare species, they can be saved. Okay, rare and very really endangered species, we can keep this thing safe. Okay, because we can keep on growing them again and again, again and again. But then, guys, the next thing, by using this technique, tissue culture technique, guys, we can bring about something that is called as somatic hybridization. Okay, so what is this somatic hybridization? Guys, from this, we are going to get some hybrids which are called as your somatic hybrids. Or the other name for this is hybrids. Okay, they are known as your hybrids. But now, by doing this thing, guys, we can get new varieties of plant. How? I will just show you. Okay, so for example, guys, what do we do? This is known as your protoplast fusion. Okay, somatic hybridization. This is also known as protoplast fusion. But guys, what is this protoplast? Protoplast is nothing but when the cell wall is removed, okay, from the plant. That is called as a protoplast. Okay, so in this protoplast, guys, now what are we going to do? We are going to take two cells of your own this thing, and you are going to just fuse them in the presence of PEG. Okay, two protoplasts of your desired plants, and you are just going to fuse them in the presence of PEG. PEG is basically polyethylene glycol. Okay. And now by doing this method, guys, what have we got? We have got one. First one, by the fusion of potato, okay, potato and tomato. Now what have we got? We have got tomato. Okay, and second one, guys, we have a Raphanus and we have your Brassica. Raphanus plus Brassica. This is going to give us a Raphano Brassica. This is Raphano Brassica, guys. So this is what we need to know with regards to somatic hybridization. Okay, but now, guys, the next part that is there, it is a little small part, but it's a little big as well. Okay, so what we will do is we will stop over here for the day. Next class, when we come back, guys, we'll finish off with that small part of your single cell proteins, and then we'll do some question and answers related to this. Okay, Tissue culture is also used in ornamental plants. Yes, 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 yes. It is going to be used overall. See, it is very easy to cultivate plants by this method. Okay, so that is why we will go for this kind of thing. Like it is easy, cost effective, and plus, it is season free, disease free. Okay, by this we are producing mangoes now, actually. So that way. Okay, so guys, once again, what we will do is we'll stop over here for the day. And once again, I'll see you in the next class. Till then, take care. Bye bye, everybody. Bye. Bye bye. Bye.